Well, good morning, everyone. I'm going to try again. Good morning, everyone. Good. This way I know you at least start off awake, and then we'll see what happens. So I was very excited to be invited to be part of this panel on harnessing information for HIV prevention. It's rare to see a combination of social science and sort of deep mathematical science on the same panel, and I am also eager to see how it all comes together at the end. So my task this morning is to talk about syndemic theory and how it can be harnessed for HIV prevention. These are my disclosures. And I'd like to start off by acknowledging the people living with HIV who've shared their wisdom, their expertise, their bodies for the purposes of research, and that we could not be where we are today without their contribution. So maybe some of you in the room came because you are in love with syndemic theory, but I'm guessing many of you came because you were like, what is that word? It sounds pretty cool. In its most simple form, a syndemic means a synergistic epidemic. It is a term that was coined by Meryl Singer in 1996. It has shifted depending on the context in which it's used. It has a sort of purely biological definition that's sometimes used in the medical literature as a set of linked health problems that involve two or more, and affliction are in quotes, interacting synergistically and contributing to excess burden of disease. But the social science definition and the definition that's been used in HIV research has been the concentration of deleterious interaction of two or more diseases or health conditions in a population, especially as a consequence of social inequity and unjust exercise of power. This is the most recent sort of bringing together of syndemic theory that Meryl Singer published in 2009. The theory emphasizes the role of social and cultural forces in producing epidemics. It's different from interacting epidemics. So if you think about TB, HIV as two epidemics that interact together at the biological level, syndemic theory goes beyond that. It's not the same thing as comorbid conditions like having depression and substance use because it requires a synergy. And it's more than multiple risk factors added together, although that's often how we see it operationalized in the literature. There are three key elements to syndemic theory. The first is disease concentration. So two or more diseases have to concentrate in a particular social context. So either a population, and in my work that's often key populations, or in a geographic setting. The second is disease interaction. And this is what's often left out in how people operationalize and analyze syndemic research. It's interactions between epidemics, both at the individual and the population level, that amplify the disease burden. So that interaction is quite important. And the third is also surprisingly left out is large-scale social forces. So harmful social conditions drive the disease concentration and the synergistic interaction. So those three elements are what are required for sort of classic syndemic theory. SAVA, which some of you may have heard of, substance abuse, violence, and AIDS, is sort of the seminal syndemic that was first described in the literature by Meryl Singer in 1996. Not surprisingly, it was studied among women and girls. And recently, I think this was published in 2016 or 2017, Louise Gilbert and colleagues published a systematic review looking at the literature that had been accumulated to date on the SAVA epidemic among women and girls. And you can see there, it says there remain significant methodological and geographical gaps in intervention research in the SAVA epidemic. And in fact, I'll get to this later, but there are huge gaps in intervention research related to syndemics overall. And that their global review underscores the need to advance a continuum of multi-level integrated interventions that target the mechanisms of SAVA epidemics, especially for adolescent girls, young women, and transgender women who use drugs. Since that seminal SAVA syndemic was described, many other populations have explored how syndemics influence HIV in that population. So this is one example by the National Minority AIDS Council that was adapted from Haladkis and colleagues of a biopsychosocial drivers of syndemics among men who have sex with men. And you can see it, I don't know if I have a pointer. Yeah. So you can see it includes these multi-level, as a model, includes these multi-level influences. So biological influences, behavioral influences, psychosocial and structural influences. So things like minority stress, but also social capital, safe schools, and legal protections. And all those things together influence the syndemic conditions that interact with and exacerbate HIV. We've also seen a growing literature on syndemic conditions among transgender populations. So our team did a global epidemiologic review of HIV infection among transgender populations, and we wanted to look at how much work had been done among syndemic drivers of HIV in transgender populations. And we found actually very little. We found only 8 studies that really looked at syndemics, and most of those looked at syndemics on the individual level, not at the structural or social level. And this is more than, I think, 10 years after Don Operario described sort of personal experience and from his research the experience of syndemics in trans communities and how there was a need for multi-component interventions to really address those syndemics. So even with these gaps in literature, we do see efforts to address syndemic conditions in interventions in a multi-component way. So this is a slide that describes an initiative by the Health Resources and Services Administration of the U.S. federal government that took place between 2012 and 2017. So they provided funding to 9 community health centers and community organizations around the United States to implement multi-component interventions to improve engagement of transgender women of color in HIV care, transgender women of color living with HIV. And you can see they had a variety of different interventions undertaken by these 9 organizations, from community outreach to peer navigation, individual behavioral change sessions, HIV testing, motivational interviewing. But what you might notice is there aren't structural interventions explicitly listed here. More recently, there was a systematic review of the syndemic literature that looked at, well, what do we know about syndemics so far? So there were 71 quantitative studies that were published through 2016. 75% of them were in the United States, although this trend was decreasing over time, so more and more global research taking place. And the majority focus, surprising to me, was on men who have sex with men, given that the beginning of the syndemic description in the literature was SAVA, which focused on cisgender women. Psychosocial conditions were most commonly studied, with more than 80% of studies looking at mental health, substance abuse, and violence. And the outcomes were not HIV or STIs or other clinical diseases, but HIV risk behavior in 63% of the studies. And all of these studies analyzed the data at the individual level. None of them had multi-level or systemic or social level analysis. 41% of the studies examined additive associations. So instead of looking at interactions, they looked at what happens if you add risk factor after risk factor after risk factor. However, that trend is also decreasing over time. And there were 0 studies of interventions. In fact, 79% of the studies that used the term syndemics were cross-sectional, and only 21% were longitudinal, making it hard to make causal inferences, right? So what I did find, I was very excited to find one study that was actually a publication of a protocol that plans to use syndemic theory. And I found it very interesting because we're in Amsterdam, and this study is planned in Amsterdam and might well be ongoing right now. So if the person who's doing the study is in the room, I'd be excited to hear from you during the discussion time. And this study is looking at men who have sex with men with high-risk behavior. So they plan to enroll men who have sex with men who are living with HIV and had had an STI in the last 24 months, or who are not HIV positive but have used PEP or had an STI in the last 12 months. And they plan to compare the standard of care group, which is motivational interviewing, STI screening and treatment, and linkage to co-located care, which is part of sort of the multi-component intervention. And they're going to compare that to other men have those same interventions, but those men will get an additional questionnaire that addresses the syndemic domains and then specific feedback on those domains. And you can see the domains listed there include addiction, anxiety and depression. I had to look up allothymia. I didn't know what that is. It's apparently a personality trait that doesn't allow you to understand your own emotions. I learned something new. Partner violence, childhood sexual abuse, and discrimination. And then the outcomes that they were looking at is whether the participants looked for help, sought help for their addiction or mental health problem, and the secondary outcomes were STI incidence and risk behavior. So this is a very complex, well thought out study, but you'll see it also has the challenges of outcomes that are not necessarily related to disease, although STI incidence is a secondary outcome, and then it doesn't look at those social and structural forces that might drive the syndemic domains that are being examined. So in short, I was asked to describe what interventions are out there. Not much. Unfortunately, incomplete operationalization of syndemics, so not looking at multi-levels, not focusing on the structural drivers, result in a failure to implement and test syndemic interventions. multi-component interventions are important, but they often fail to address those social forces beyond the individual level and are often limited to one disease or one condition. There are challenges in implementing syndemic approaches, as you can imagine. It requires systems thinking, and I work in an epidemiology department, so we were trained in like, this is the risk factor and this is the disease outcome, right? And system thinking requires us to move beyond that, and most of us don't have experience in doing that, and it often, depending on where funding is coming from, there may not be political will for those types of systemic changes. And measuring syndemics and quantifying their impact on synergistic social and biological systems is complex. I could have written a whole talk on the complicated ways that people have attempted to try to address those three key elements of syndemics, particularly the disease interaction component, is hard to quantify. But I think that thinking about HIV and its related syndemic conditions in that framework is very helpful to us. It reminds us to focus on social and political forces that drive the structural vulnerabilities that we see, as opposed to sort of the risk factor mentality, which sometimes ends up blaming people living with HIV for having HIV rather than looking at the social conditions that drove their risk to start with. It identifies inequitable distribution of power as the source of inequitable distribution of disease. And when it's fully applied, it can promote interventions at the policy as well as the clinical and individual level. So thank you for your time, and I look forward to our conversation.
Thank you so much. That was really absolutely fascinating and brought us all up to speed on the theory. So it's now my pleasure to introduce Christoph Fraser from Oxford, who's going to give us some information on understanding infection clusters and whether this can help us to target prevention. Christoph.
Thank you. Okay, so I'd like to start by thanking the organisers very much for inviting me. So I'll be talking about phylogenetics and how we can generate information that might help us in designing targeted prevention. So I'm representing the Pangea HIV Consortium, funded by the Bill and Melinda Gates Foundation. So we have 6 epidemiology research centres. So these are centres with very well-characterised populations. Some of them are doing test and treat trials. A lot of them are doing field epidemiology. We've so far generated about 15,000 HIV viral genetic sequences, and we're going for 30,000, and our aim is to understand the heterogeneities in the epidemic and infer sources, sinks, and hubs of transmission. So I just want to step back and see where we are with DNA sequencing. Many of you will know this, but DNA sequencing is becoming ubiquitous. It's everywhere. With portable DNA sequences, you can see people sequencing on a glacier. You can see sequencing in space. And in the bottom right, you can see somebody sequencing a Zika virus very accurately in a car park in Brazil. The data were accurate enough that they were published in Nature. Next generation sequencing is coming to the HIV clinic. This is a BSL-3 container lab in Lusaka in Zambia, and this is our Oxford team just bringing the protocol. There are 2 sequences on the desktop, one of them sequencing HIV, one of them sequencing tuberculosis. For a high throughput facility, it's still the case that it's easier to do that in a centralized lab. This is a setup in our Oxford lab where we can do 384 samples a week. That's with a single technician and a bunch of robots. The protocol has been optimized over a couple of years by David Bonsall, who will be giving a presentation at 8 a.m. The current cost is £30, or about $45 per sample, and it's a single assay which generates a viral load. It gives you the drug resistance data with all of the minority variants, including short minority haplotypes, and can be used for epidemiology. One of the things that's not talked about a great deal in next-generation sequencing is that the lab work is only about half of the work. a lot of the work is computational in terms of interpreting the data outcomes. It can be made fully automated, but actually if you want to make it fully automated, it's quite intensive, and we do it in a secure high-performance cluster you can see on the right, but a clinical application would require a cloud-based solution. So one way or the other, unless we solve this computational hurdle, we're talking about data moving. So data sharing at some level is implicit in the application of this technology. Why do we sequence HIV? What do they add to epidemiology? So if you think about classical epidemiology will be in a cross-sectional study or in a cohort study. You'll identify the people who are or who become HIV infected. That study design has proven very powerful. When done in couples, it's shown us that ART in the HIV-positive partner is the most effective intervention in terms of reducing transmission, as well as reduction in susceptibility due to male circumcision. It's essentially the basis of all of our knowledge that we've translated into prevention. When we genotype the virus, we compare the RNA or the DNA code of the viruses, and we essentially look at which people have more similar viruses. So we cluster together individuals in terms of those who have similar viruses. In this case, the 3 who have the green virus, the 2 who have the red virus, and the 1 who has the blue virus. And we can ask, those people are probably related by relatively short chains of transmission. We typically don't know anything about missing intermediates and such like, but we can ask, what are the assortative traits of infection, or in other words, are there sort of sub-epidemics? So to give you an example, these are about 60% of the cases amongst MSM with subtype B infection in the Netherlands here, and each horizontal line at the top is a separate cluster, with each dot being a patient. And in the middle band, you see the small clusters, and at the bottom, people who don't cluster. When we ask people who don't cluster, they're much more likely to report having acquired infection abroad, outside of the Netherlands. You can see separate clusters for heterosexuals and MSM, and you can see a giant cluster of green and red, which contains many injecting drug users, and also that's essentially where almost all of the hepatitis C virus co-infection lies. We can look at the different clusters, and they have properties. The first is that despite 20 years of prevention, not a single one of the sub-epidemics was brought to a halt. And the second thing is that they actually have very homogeneous dynamics. So this is sort of a measure of how much they're spreading. The spread went down in the 1990s and sort of increased in the first decade of the 2000s. And there wasn't a great deal of difference between the clusters. There weren't very rapid clusters and very slow clusters. The third thing, as you see in green, is that the most recent clusters are the ones which are growing the fastest. And if we look at the defining characteristics, the defining characteristic is age. Most of the older clusters are increasingly spreading in older and older men, and the more recent clusters are the ones which are taking off in the younger men. What could we do next with epidemiology? The next thing we can do is we can try to resolve the direction of transmission. If we were to do that, so in this case I've now got directed arrows rather than clusters. If we were to do that, we could ask what the characteristics are of a source case, and how can we better target prevention. Now, one way of doing that is if you have data on the likely date of infection. You can either have that because you have very good data monitoring, as is happening here in the Netherlands and in many other countries, or you can infer it through various methods of back calculation. There are many uncertainties when you do this. You typically have an uncertain date of infection. When you draw these arrows, you don't know if there are missing intermediates, and it's actually quite hard to rule out the possibility that there aren't common sources to the cases. Those uncertainties are important if you're thinking about this from a sort of forensic basis, but actually for epidemiology, you only need to be right most of the time in order to derive epidemiological covariates. So here I've shown likely dates of infection for the individuals in this graphic. So we did this precise analysis in the Netherlands again, where there was very detailed clinical monitoring, and we found for each recipient who was a seroconverter, we found one or more of their probable transmitters, in some cases putting different probabilities to the two. And we looked at the time when infection was likely to happen, what clinical stage the transmitters were in. And we found 883 such transmission pairs, and we classified the probable transmitters into one of 13 possible clinical stages. In the publication, we give the proportion of transmission attributable to all of these stages, but here are the sort of headline figures, is that nearly 70% of transmission over sort of the 15 years studied occurred from undiagnosed individuals, and about 40 to 45% from individuals in their first year of infection, and that very few transmissions occurred from people who were on ART. And remember, these are the early days of ART, when people had frequent blips or issues of adherence. Next, I just want to move on to a new way we've developed to infer the direction of transmission. And the insight here is that when we sequence, we don't have to limit ourselves to sequencing one virus. We can actually sequence the whole cloud or quasi-species, or just a diversity of viruses which are in individuals. And if you imagine two people, in this case person A and person B, who each have their cloud of viruses, we can reconstruct the ancestry of those individual viruses. So this is now the evolutionary history of the viruses, not the transmission chain. And you can see the 4 viruses from person B, shown in red, have an ancestor which is here. But when you reconstruct the ancestry of person A, the ancestor of all of the viruses at this point, this is time going forwards. The key point is that the ancestor of the virus from person B lies within the diversity of the viruses in person A. That suggests strongly that person B was infected from person A. You can't rule out missing intermediates, and the longer this amount of evolutionary time here, the more likely it is that there are missing intermediates. We formalized how to do this with next-generation sequencing using our software Shiver and Phyloscanner. And the way that works is, as standard, this assay, which I talked about that costs £30, sequences many fragments of the virus. We typically get millions of these fragments, the DNA sequences. And we look at where they are on the genome. And then at any one point in the genome, we take a slice in the genome and we take all of the little fragments of viruses and we work out their ancestry. And you can see that in this case, if the blue virus is, its ancestor is within the red virus, we infer that red was a source of infection for blue. This is looking at real data, and here are 4 slices through the genome. You can see that the virus from person F lied within the virus from person E in this window. Here's the virus from person F in the person E in this window, and again in this window. So these are different parts of the genome telling you exactly the same thing. You can see other fun things like person B was dually infected, for example. So we summarize this information. So this is what it looks like in one genome window. So this is now in 54 genome windows. And you can see that in 54 genome windows, 41 of the genome windows supported transmission directly in this direction with possible missing intermediates. 9 of them, well, actually 11 of them supported a direct link, but we couldn't say anything about the direction of transmission, and 2 of the fragments of the genome actually pointed in the opposite direction. So the way we summarize this is we say 100% of the windows supported transmission, with 76% of the windows supporting direction of transmission in this direction. And it's important to emphasize uncertainty. Here's a more complicated transmission cluster, but when we summarize it, it actually comes as fairly straightforward. The second number is the support for a transmission link with possible missing intermediates, and the first number is the support for that direction of transmission. If we have very low confidence in our direction of transmission, we don't point an arrow, we just say there's a possible link there. Here are the kind of things that start to happen when you do this at scale. So now let's go back to Pangea and use that to answer some epidemiological questions. So these are the 6 research centres which are part of the Pangea Consortium: RACI, the Partners in Prevention, MRC Uganda, partners, again, Zambart, and the Africa Health Research Institute. There are 2 zones, really, of sampling. There's the East African zone and the Southern African zone. In the East African zone of relevance, HIV is very heterogeneous in terms of the prevalence. And you can hardly see this, there's concentration around Lake Victoria. One of the partners is the RACI cohort, or the RACI Health Services Research Centre. These are all the different sampling sites. There are sampling sites on Lake Victoria where there's very high prevalence. There are rural sampling sites and trading sampling sites that are along the road. So with Kate Grabowski and Oliver Ratman, we applied this method of using within-host diversity to find transmission pairs, and we find 500 out of 2,652 individuals who we sequenced, out of 5,142 who were HIV positive in that round of the survey, we found 554 likely transmission pairs. These made up some transmission chains. In one case, a very long transmission chain. Most of the cases were just transmission pairs. So the next question we asked is, how reliable are this inference about the direction of transmission? Well, there were 71 cases in which we could independently say that one of the directions of transmission was very unlikely. That's when we were looking at in pairs where one was initially seronegative while the other was already seropositive in previous rounds of the survey, and the other cases where one individual had a very high CD4 count and the other individual had a very low CD4 count, where transmission is much more likely from that with the low CD4 count. And overall, we were wrong 16.3% of the time. And this is the sample where we had the most difficulty with sequencing compared to the others. But it gives you an idea of the sort of false positive rate. It's probably good enough for epidemiology, but not good enough or shouldn't be used for forensics. On Thursday, if you're interested, you can see an application where we looked at all of the transmission pairs on the fishing communities and the surrounding area. And what surprised us is that actually none of the, or only 2 or 3 of the transmission pairs link together the fishing communities, which have this extraordinary high HIV prevalence, to the surrounding communities, really indicating that these are separate epidemics. But I would refer you to the talk on Thursday. This is my last slide of results. What we did is we looked at the risk factor for whether transmission was coming from the male or whether the transmission was coming from the female. The reason this is a good study design, it's not an obvious one, but the reason this is a good study design is that these analyses are quite frail to sampling bias, and this really inherently corrects for a lot of the sampling bias because they affect the males and the females in a similar manner. So the first risk factor is viral load, a very well-known one. If the male has greater than 10 to the 4 copies per mil, you're much more likely that the transmission came from the male. And if the female has a high viral load, you're much more likely that the transmission came from the female. If it's more likely from the female, I put it on the right, and if it's more likely from the male, I put it on the left. Next is a number of partners, self-reported. It wasn't significant for women, but it was significant for men. If the man reported more than one partner, you had an odds ratio of 1.78 for transmission from the male. The age effects are very interesting. If the male was involved in transmission, he was much more likely to be the transmitter if he was aged 30 to 34 than either if he was younger or if he was older. So this is the age group where the male was most likely to be identified as a transmitter. That's interesting because that's the age where many men are still not linked to care and are still not on ART. We know that from population surveys. Conversely, if the woman was young, less than 25, and the direction of transmission was also much more likely to come from the male than if the female was older than 35. So again, young women are much more likely to be the recipients of transmission than older women. In terms of sort of some indicators of vulnerability, if the female had no education compared to at least primary education, or was a recent immigrant into the community compared to a previous resident, she was much more likely to be the recipient rather than the transmitter of infection. These are the odds ratios. The first one, I guess, is borderline significant. If the couple were cohabiting, so we did the analysis, blind to whether they were cohabiting. If the couple were cohabiting, the male was more likely to be the transmitter of the two, with an odds ratio of 2. And finally, the one exception to this was if they were both in a fishing village, in the same village, then the female was more likely to be the transmitter. So to summarize the epidemiological finding, with next-generation sequencing in a single assay, it's possible to do source attribution. It's not 100% accurate. It's about 83% accurate in this population in calling the direction of transmission. The fishing communities were epidemiologically separate from inland communities, and that's relevant because there's been a policy of targeting the fishing communities for enhanced prevention, even though they're very small, and the hypothesis that they would have a disproportionate effect. Male to female transmission is more likely in households if the female is young, uneducated, or an immigrant, or if the male is aged 30 to 35 and reports more than one partner. Female to male transmission occurs more often outside of the household and in the fishing communities. Next generation sequencing has allowed us to identify hundreds of transmission pairs and chains. And with many applications in HIV epidemiology, there's also the opportunity to understand the biology of HIV transmission. You can look at what are the properties of the viruses which are very close to transmission compared to the other viruses. And I think we just need to be ready, linking to the last talk today, for the fact that this is going to be cheap, accurate, portable, and very difficult to do without moving the data to clouds, be they local or international. Oops, wrong direction. Thank you. That's the genome of DNA of HIV. And those are my acknowledgements.
Thanks, Christoph, for that fantastic talk. My name is Roger Kouios from the University of Zurich, but you're lucky enough that you don't have to hear me speaking. Rather, we'll introduce the next speaker, which is Frank Tanzer from the Africa Health Research Institute and the University of KwaZulu-Natal in South Africa. And he's working on evaluating and designing intervention to curb HIV transmission, mostly using geoinformation systems and also partly epidemiology. So thanks so much, Frank.
Okay, good morning, everyone. I'm going to be speaking on the use of spatial analytical approaches to identify ongoing HIV microepidemics in rural South Africa. And I'd just like to start by thanking the conference organizers for inviting me to give this presentation. It's a great pleasure to be here and to be able to deliver this address. So I want to give you a brief outline of my talk. I'm going to start with an introduction to orientate you to the social and the epidemiological context of where we're working. Speak about HIV microepidemics that we've identified, and then speak about some recent examples of work that relate to those microepidemics in terms of population viral load, geography of transmission, and finally how these are all feeding in to the design of an intervention. So by way of a brief introduction, we know that there's been this fantastic scale-up in ART globally. And South Africa has the largest program in the world with now 4 million people on ART in 2017. And our study area is located in the northeast of the country of South Africa in the province of KwaZulu-Natal. And that's considered by many to be the epicenter of the epidemic. And we've been privileged to be positioned there as ART has scaled up and to measure some of those resultant impacts. So this is now looking at the study site in detail. So the Africa Health Research Institute has, since 2004, conducted population-based HIV testing. So in that grey area that you see there, There's 90,000 people living. Every adult in that population is eligible for population-based HIV testing. So we have field workers going around, knocking on doors, consenting people to test for HIV via a finger prick. Those red crosses that you see there are ART clinics, primary healthcare ART clinics, where ART is delivered by trained nurses and counsellors. And that area is about 400 kilometers squared. It's a very poor rural area. It's in the second most deprived district in South Africa. And bordering this population-based HIV surveillance is the treatment and prevention trial area. I won't be talking about that today, but just focusing on that grey area, which is 20 by 20 kilometers. So as I said, this is a deprived setting. It's mostly rural, but there are pockets of urban and peri-urban populations, and I will be alluding to those in my talk. The adult HIV prevalence is sitting at around 29-30%. So it really is a very hard-hit community. So we know that ART is having a profound impact on the epidemic. Since the rollout in 2004, if you look at the top graph there, you can see the spectacular increase in life expectancy, increasing from round about 50 years of age at rollout, this is in adults, to over 61 years of age in 2012. So more than 1 year of life gained across the entire adult population for every year post rollout. So this is one of the largest impacts of a public health intervention documented. Coupled with that, this very exciting finding published in Science showing that high coverage of ART in a community reduces an individual's risk of acquisition of infection, even when delivered through an imperfect rural program such as we have in this setting. And as we move towards attaining and hopefully exceeding the 90-90-90 targets, there's a real opportunity to monitor those resultant impacts in this type of community. And our overarching question is, how can we interrupt HIV transmission, especially given the high incidence rates that I'll show you just now in this era of access to ART. And the key questions within that are, what is the best overall combination prevention approach? Secondly, where should we target? And thirdly, who should we target? And for the basis of this symposium, I'm going to be focusing mostly on where should we target. So now I want to speak, having given you a bit of introduction to the epidemiological context, I now want to speak about HIV microepidemics. And here are 3 papers which looked at the geographical heterogeneity in the epidemic. And if there's one message I want to get across today, it's kind of the extraordinary heterogeneity that is available in the epidemic. And that presents, I believe, a real opportunity to intervene in the most vulnerable communities for epidemic control. So if you look at the map on the left, that is HIV prevalence. You can see that it varies from less than 10% to greater than 35%. Those circles that you see there are formal clusters of infection, of existing infection. So the 3 overlapping circles along the national road, those are clusters of high numbers of infections that are unlikely to have arisen by chance. This is using a Kuldorf spatial scan statistic. The smaller blue circles are unusually low numbers of infections that are unlikely to have arisen by chance. So these clusters of high infections are peri-urban communities along the national road. They are characterized by high numbers of lifetime partners. They're very mobile populations. The map in the middle shows all-cause mortality. You can see a cluster of high numbers of deaths in those same communities. The map on the right then shows incidence, HIV incidence, population-based. You can see the variations ranging from less than 2 new infections per 100 person years to 4 and up to 6. And those ellipses there again represent unusually high numbers of new infections with a relative risk of 1.9 and 1.44 respectively. So all identifying these same kind of peri-urban communities as being really vulnerable to all these HIV outcomes. And we did this review with several colleagues, looking at kind of concentrated HIV sub-epidemics within generalized epidemic settings. And what we find is across Africa, there are these concentrated epidemics within generalized epidemic settings. So it's not just a local phenomenon. Now, one of the things I want to mention is heterogeneity across space, but also by gender. So one of the greatest challenges in HIV today remains this incredibly high burden of new HIV infections in young females. So if you look at the graph on the left there, you can see incidence peaking at 8 new infections per 100 person year. So just outrageously high in this community. And one of the things we think is going on is that it's these young men with high viral loads who are not accessing care, who are transmitting to these young females. We've got phylogenetic evidence to back that up. So it's really critical that we find a way of getting these men tested and into care and onto therapy. And I'll be speaking more about that as we go along. Now, this is some recent work taking the clustering of incidents further. And if you look at the map on the left there, we're using a technique here called a Tango's flexibly shaped scan statistic, which doesn't rely on a cluster being circular or elliptical. It can be any shape. And what is interesting in this paper is that we found these two clusters along the national road. And in a sense, that's exactly what we expect. There's no surprises there. But what was interesting, we found a third cluster near a recent coal mine development. With an incidence, a population-level incidence of greater than 10 new infections per 100 person years. It's among the highest sort of population-level rate we'd ever seen. Cordelia Coltart presented some results at CROI where she took this further and looked at, if you look on the right there, a phylogenetic cluster of 75 linked infections. A large proportion of those were male, about 60%. With two kind of bursts of infection in 2012 and 2013, respectively. And the mine itself started operations in about 2010. A large proportion of these, 40%, clustered immediately around the mine, another 40% in those peri-urban clusters. And this was just an illustration of how rapid economic development can lead to unintended health consequences. And it's really necessary that the health services provide interventions that are adaptable and able to respond to this kind of issue. Now, of course, we cannot attribute causality to the mine, but it's strongly suggestive of a link. I'll speak a bit about the population viral load work. So population viral load, or aggregation of viral loads in a specific community over time, has the potential to provide a very sensitive pulse on the epidemic. And what we did in this study was to take a population-based viral load survey, look at the viral load patterns by community, and analyze the extent to which that predicted future incidence. This paper published in Science Translational Medicine. And if you look at the map on the left there, now I've rotated this 90 degrees for better visualization, but that map shows the population prevalence of viremia. So that is of HIV viremia. So that is the proportion of the entire population, again, not just amongst the positives, this is of the entire population that is viremic for HIV. So in those hotspot communities, so-called hotspot communities circled there, 25% of the entire population is viremic for HIV. Again, not just among the positives. So if I reach into one of those communities, take someone out on average, there's a 1 out of 4 chance that that person will be viremic for HIV. And I think when one is confronted with statistics like that, one begins to get a real sense of the magnitude of the problem we're dealing with here. But the map on the right, the graph on the right then shows the relationship between the population prevalence of viremia and HIV incidence. So it's by quartile, lowest population viral load quartile, highest 1 to 4. And you can see a very strong relationship to incidence. That is an ecological relationship, but when one does it at an individual level and controls for various risk factors, that relationship holds. So there's a lot of potential for these sort of indices in terms of identifying the most vulnerable communities and intervening in them. What is really striking in terms of the viral load, and again, this is population-based. This is not people who are attending care. This is population-based field workers going around testing from door to door. What is really striking is that you get this massive gender differential. So when women become pregnant, that is their link to the healthcare facility. They then get tested and put into ART. There's no such point of entry for men. And so you see there's a far higher viral load among men. So given that viral load is higher or lower among women, given that circumcision is scaling up, what we've seen is this strong drop. What we've seen is this strong drop in HIV incidence in men. And this is featured in the UNAIDS 2018 report. I'll refer you there if you need further detail. But sadly, no such reduction for females. And so again, this highlights the fact that we have to get these men onto care. We have to get them treated. Now I want to move on to speaking about the geography of transmission. So when we think about these communities, a really intriguing hypothesis becomes, to what extent do these high-risk communities, or to what extent are they actually the engines of the epidemic? And is it possible that they're reseeding micro-epidemics elsewhere? And if so, is there a real opportunity to intervene in those communities and have a big impact on the overall epidemic? And we can test this hypothesis with phylogenetics. And this is some of the work presented by Diego Quadras earlier this year at CROI. But if you look at the map on the right, so each line here represents a linked infection. So that is 2 individuals linked by the genetics of the virus. And there's been an intentional random error introduced, so these are not the exact locations, obviously. But what you can see is that a significant proportion of those linked infections seem to hark back to the hotspots. So giving some initial support to our earlier hypothesis that these are actually responsible for reseeding microepidemics elsewhere, and therefore we could have potentially a big impact if we were to intervene successfully in those communities. So I guess the question is that after having shown you this, how does that then feed into an intervention? And one study that I'm very excited about that really speaks directly to this I'd like to present now. And this intervention really seeks to address 2 of the main issues in HIV today. And that is the high level of new infections in young females and the high level of male HIV-related mortality. And both of these are actually 2 sides of the coin, and just means that men are not accessing care, and we have to find a way of doing that. So this is a cluster RCT with a baseline adjustment for factors going backwards, and I'll speak about that now. But it's essentially a 3-pronged approach. Firstly, it's a financial incentive to rapid test at home. There's an ability to earn a second financial incentive by linking to care. It's accompanied with some gender-sensitive counseling as well. And the beauty of this sort of approach is it leverages the statistical power going back a decade to be able to detect a very small drop in HIV incidence. So it's powered to detect a 25% drop in young female incidence, 15 to 30 years. Other outcomes are male viral load and male HIV-related mortality. So that has just started, and the results, the intervention will be finished being delivered by the end of the year, and then the follow-up will take place for another 3 years. But we're very excited about that. And really the issue there is to try and get the population viral load down in men, particularly in these high-risk communities, so that they do not infect females, and so that their male HIV-related mortality, its unnecessary deaths, are averted. So conclusion, I hope that's given you an idea of the sort of micro-epidemics that exist in our setting, and the analyses suggest that exploiting heterogeneity and risk, be that geographical or otherwise, is key for epidemic reversal. And so we need to take a precision public health approach to ensure that no one is left behind, and that means tailored interventions to reach the most vulnerable groups. Our initial findings suggest that the so-called hotspot communities are also playing a substantive role in reseeding microepidemics in other areas, and so novel interventions targeting high-risk groups and geographies are really critical. So acknowledgements, I'd like to thank the funders and many people who helped with this. Also thanks to the members of my team who also contributed in a lot of ways to this presentation. So thank you very much.
Thanks, Frank, for this fantastic talk. I think we have heard now a very fascinating combination of methodological approaches, both theoretical and from the point of view of data analysis and for the intervention. And what we want to do for the last talk, given by Gail Henderson, is step back and think and discuss and think about the ethical, societal, and legal implications of these technologies. Thanks so much. Yeah.
Good morning. Okay, you're still awake. Although I have to say, I'm so fascinated by those talks, it's hard to imagine not being awake. I realize I have neither added a slide about conflicts, I have none, nor have I added a slide about acknowledgements, and I guess I acknowledge everyone I've ever worked with. So I have the really simple task of trying to summarize ethical considerations that may arise in these different and sometimes really quite innovative new technologies that are being used to harness information for HIV prevention. And in this talk, I really was planning to focus on pharmacogenetics, since it is the genetics of the virus and the new technologies that are being diffused to get ahead of prevention efforts. So, or as I think Frank said, precision public health that I want to focus on. But I have a couple of questions for the other presenters, and of course I'm going to try and do this quickly because I'm sure there's a lot of comments from the audience. One thing I want to point out is that this new field of ethics and HIV phylogenetics is quite new, and it's generated several recent publications. I want to point out that two of them are going to be presented as posters. Kumi Smith, Ethical Pitfalls of Network-Informed Approaches to HIV Prevention, is a general ethical consideration, but she has embarked on work on public health HIV prevention work using phylogenetics in North China. So you can stop and ask her about that work. There's a multi-authored paper that's just in press that came from a meeting in London hosted by the Gates Foundation about ethical considerations on global HIV phylogenetic research, another one from the US CDC, and finally one that applies a very frequently used model in clinical ethics. So I've relied on these to a certain extent. But I think it's obvious as you've listened to these talks that the basic challenges in HIV phylogenetic research and in the kind of research which really focuses on the targeting of individuals through these kinds of new technologies is relational, that it's not just about an individual, it's about an individual and partners. And it may be also about an individual living in a community. And the example that Christoph provided with the Fisher villages that were highly stigmatized, had a huge epidemic, but apparently not contributing. And Christoph, am I right about that? That's what you said. So this was in a sense a kind of liberating scientific conclusion for the community. But in other cases, it could have the opposite effect. It's people, it's their partners, it's these hotspot communities that they live in. And the second quite important challenge is that real, true anonymization of viral sequence data is difficult and may not be possible, particularly when the sequence exists through another study. So Those are two key issues I want to point out. Then I want to turn to 5 different aspects of good ethical practice, and to me they raise questions that I'd like the speakers to address in their own work, but I would like to hear comments from the audience as well. I mean, one of the most important things, and I have to say, when I had friends say, should I come to this part of the presentation, I said, no, you actually know all this. We all know all this, so we'll see what you think. But balancing individual risks with these new technologies and the clear goal for public health and scientific benefits is always a tension. And as I demonstrated in the comment from the Rakai site, protecting individual and group identities is going to be very, very important to individuals who may or may not be consented, communities who may or may not be consented to participate in this kind of research. There's a number of serious social and legal contexts. This is all taking place across the world now, but certainly the rural South Africa context, the context of the 6 sites that Pangea is implementing its work, compared to those sites that are now using these technologies in the US, are going to generate different concerns, particularly if we think about criminalization of behavior that leads to an HIV diagnosis in some parts of Africa. And of course, the kind of safeguards we would want for this new work include informed consent and community engagement. And I mentioned who really will be asked about this. And one question I had from Christoph's talk was, did you, for those sites, how are people being consented for that work? And I think for Frank as well. And then finally, equitable data sharing. So the funding for phylogenetic work, you saw the fancy central laboratory, which looked awesome, $45 a sample, all the genetics you ever wanted to know about all the viruses somebody has. But then there'll be decentralized labs. Still in all, the release of those data to centralized labs raise critical questions about equitable data sharing, about authorship, about how people can be assured of the protection of their identities. So, and my thought was a lot of this is not new in HIV prevention research. We've talked for many years about exposing individuals to the risk of stigma or to legal jeopardy, protecting confidentiality, anonymity, employing community consent, which is something that's being rolled out in a variety of places for this kind of new technology. It's not new. But what's new and which I referred to previously and raises questions about, how and when you actually consent people in communities to do this is that you've got these people implicated through phylogenetic transmission networks and their communities, their partners, et cetera. And yet you have the challenge of explaining to somebody, okay, I'm 76.3% sure that this is the direction of the transmission. So it requires, all of you, I'm sure, understood the talks perfectly, but possibly the communities in which these technologies are rolling out might find it more challenging to really understand the genetics of the virus and how these technologies are and the probabilities of the direction of transmission, that one person in the middle that isn't being looked at that really might be the important transmitter, that's going to be quite hard to explain. And it's not clear who should be making these decisions when the implications are for communities and for individuals as well. I come from the world of a program that NIH funds. It's called Ethical, Legal, and Social Implications of Genomics. It's about human genetics, human genomics. And when I first heard about this, I thought, wow, I've got a lot to offer. because there must be a lot of overlaps in things we've worked on and the things that we're so keen on, and this is a lot of the research that I do. So the meaning of genetic information, will genetic risk be understood? What really should be returned or not? How's information relevant to family or other group members? Who controls that information? What happens if it's shared? And implications for employers, insurers, and the state, as well as the people I've already described. And then finally, for human genomics, will diagnoses have reproductive or eugenic implications? So if we think about translation to the genetics of the virus, well, it sounds pretty similar, doesn't it? The ethical and legal issues that are really have been studied a lot in human genetics. But the idea, the social meaning, and the communication about this new technology is not at all. It's novel. It's not been done. Although it's not just HIV. Since I've been here, I've learned that, for example, TB is also being subjected to the same kind of phylogenetic work. So this is going to expand in the genetic identification of all kinds of pathogens. So I think it sort of behooves people who are in these fields of ethics and social implications to really pay attention to what are the new issues. How can people relate to information about about the viruses? Do they become part of their identity? Is there more or less responsibility or blame on transmission networks? And how can communities as well as individuals be able to consent? So that's it. And I do wonder if I could. What time have I got? What time is it? Yeah. Okay. Well, it's just a question. I'm thinking about not just the questions for Frank and Christoph about, well, what happens in their studies? How do they protect people? But also, Tonya, you introduced us to this idea of a more macro level, more synergistic, what's the word? Syndemic view of how people experience disease burden. And these are at the community level, but also at the individual level. And I guess I would say, how can you link the kinds of issues that you were focusing on to these in the ground, very scientifically sophisticated new technologies that are going to help with, getting ahead of transmission networks to really improve prevention. So I leave with those questions, but I also thank you for staying awake for mine too, and thank you.
Thank you for that. Does it work? Thank you for that fascinating talk. The floor is open now for discussion and questions. I see there is already one question. The front, please.
Well, thank you. Tim Mastro with FHI 360. That was really a terrific session, and it recalls when HIV recency testing came out 20 years ago. That test that could tell someone you were infected within 6 months was exclusively used as an epidemiologic tool, and individuals were not given the result because of the possibility that if you confirm that I got infected within 6 months, then it was the partner within 6 months that was the source of my infection. This takes it to a whole other quantum of linking folks like that. So great data. So the question is for Frank. Again, thanks for another brilliant review of the data you've generated from your studies in that community. And the idea of moving towards finding men that are in that 25 to 35-year-old age group that are apparently the source of a lot of infections, getting them getting on treatment certainly makes sense. But I guess the question I have is, when these data are presented or revealed in the community, and you've sort of verified what I'm sure was common knowledge for decades, that teenage girls and young women are having sex with older men, and that results in both STIs and HIV. How does the community react to the data showing we've got epidemiologic hotspots, we've confirmed locations, and we've confirmed this the generational age difference in men and women is there a community response to that dynamic, and how a community could be part of the solution?
Yeah, thanks for that question, Tim. So we have a very comprehensive sort of set of liaison structures to liaise with the community. And we have a whole lot of feedback structures where we feedback this information. So the response so far has been one of, we're glad that we know. So in terms of the sort of geography of transmission, and in terms of the sort of hotspots, that's something that people knew intuitively. They knew that these are high-risk areas, so it was nice for them to have it confirmed. And it's been good in terms of that we've been able to interact with the health services and let them know, we now have evidence that you need to do something proactively in these communities. So that's been well received. Also, in terms of the sort of age difference thing, so the waters had been muddied by the fact there was a lot of focus on sugar daddies, unnecessary focus, it turns out, having now. So again, there was a sense of, okay, well, now we know we can do something about it, so thanks for letting us know. So they were quite receptive, and they were glad that we were able to sort of demonstrate that. And so we haven't had any negative feedback, and it's been quite a positive interaction.
Okay, great, thanks.
Hello, Andrea Wirtz from Johns Hopkins. Thank you very much for the presentations today. They were all wonderful and I think gave us a lot to think about. Gail invited Tonya to think about how this endemic approach could be mapped onto some of these phylogenetic analyses, but I would ask to open it up to all of the speakers, not just how this endemic can be mapped on from Tonya's perspective, but how Frank and Christoph might have also been thinking about this endemics, or at least the macro level factors that might play into some of these phylogenetic and geographic differences that you're seeing. Thank you.
Let's take one or two questions so that the panel can get their thoughts, and then we'll go through. Okay, the lady at the back.
Hi, Uran Green, CDC Atlanta. Thanks to all the presenters, really great information. Quick question for Christoph. I noticed your work is concentrated in East and Southern Africa, and I'm wondering what the implications would be hypothetically for applying those technologies in a place like West Africa, where some populations have up to 25% of HIV 1 and 2 co-infection. Having those two viral types in the same individual, just wondering in what implications that would have for tracing chains of transmission. Thank you.
Let's listen to Caroline's question, then we'll get some answers, and then we have 1 and 2. Okay, Caroline.
Yeah, Caroline Sabin, UCL. I actually wanted to follow up on the question from the lady over the other side, because I think the real interest in this and the real excitement is the fact you can bring together the work that Tonya's been doing, which I just, in response to Gail, I also think is scientifically sophisticated. So just one comment about that. But my question is about pragmatically and practically, how do you fund that? Because the funders are often very siloed into either the sort of more quantitative methodologies or the qualitative work or the social work. So what are your perceptions about where you would get funding? Because you would presumably need large amounts of funding to bring together all the work to develop those interventions and then test them.
Shall we go to the panel for the...
I can start. Thank you for those questions about bringing them together, because those were questions I had until today, how we were going to bring them together. It was very interesting listening to the other presenters and thinking about the social drivers behind that. And I think one example that was just screaming syndemics to me was the one about the mine and finding the new cluster near the mine and the social forces that brought that mine to that place, and then the implications for what happened to the social structure in that community when the mine was built and employed people, and how that led to what you saw downstream in the tiny little genome. So I think that there are lots of opportunities similar to that to look back at, we're not saying ignore what's happening at the biological level, but also look upstream at what's driving what we see at the biological level and bring those pieces together. And I actually don't think it's more expensive. I mean, it doesn't cost $45 per particle to collect social science data usually. So I think it would be pretty straightforward to combine that. And I see people are doing that more and more as they realize the social piece of this epidemic is critical. And I could name a number of examples, but I want to give the other panelists an opportunity to respond as well.
Okay. I'll answer first a specific question about HIV-1 and HIV-2. So in fact, that's not difficult. The assays can do multiple panels of viruses. So we can also look at the co-transmission. Typically, it's RNA viruses on one side and DNA viruses on the other. But there's no technical barrier to studying a whole panel of RNA viruses. And in fact, it opens up new avenues for looking at the co-transmission, not just of HIV-1 and HIV-2, but HIV-1 and HCV, for example, in settings where both are prevalent. Again, sort of linked epidemics. I also find the question of how do you bring together the different types of information to look at syndemics very fascinating. And actually, I think the key to that, in my mind, is bringing the right people together to be sort of interrogating the data with the right questions. But it's also solving the privacy issues. Those questions can only be addressed if all of the data are linked. And there's a lot of, I think, concerns about creating those very linked data in an area which is already sensitive from the start. So the way I think about it is you start by creating a system which people have trust in. All of the stakeholders, the participants, the community representatives, and the different agencies have trust in. And you then start linking the data within that system where people have trust. So that's going to be a long-term. To some extent, that's a much harder problem than some of the technological problems. But I think your approach demonstrates that would be well worth doing. And the question as to how you fund this work, well, I don't know. I think I push the answer back to the funders. I've been involved in one project that I'd mentioned, which is this HPTN 071 pop art trial. And one of the things that was really interesting, admittedly it was well funded from the outset, but one of the things that was very interesting is that right from the start, the funders stipulated all of the different fields that needed to be represented before you started the work. So we're all busy, but if the funder stipulates people have to get together, that tends to carry impact. So there were social scientists, there were community advisory groups, there were ethicists, lawyers, mathematical modelers, clinicians, virologists. All of those perspectives were brought round the table by the funders, and that's a powerful approach.
Just to say that I couldn't agree more with your comments about looking at the sort of overarching structural factors in regards to the mine. And the paper that I presented, which was at CROI, a paper Cordelia Coltart presented, was in fact a kind of multidisciplinary study which included a rapid ethnographic assessment of what had transpired around the mine, and included things like more truck drivers being brought into the area, opportunities for more transactional sex, although the community reported they did not see visible signs of sex work. They also said that there were more opportunities for men to spend their money, etc. So these kind of things are all being investigated, and I completely agree. Yeah.
I think you answered my question.
Shall we take the three questions and starting with the one in the front, which I think is waiting for the longest?
Yes, Mike St. Louis from CDC and PEPFAR. just add my congratulations for a really very interesting session. I have two questions. One's fairly nitty-gritty and the other's a little more overarching. The nitty-gritty one is, Frank and Christophe, you seem to find different outcomes. Your Fisher Village did not find linkage to the neighboring communities, and Frank did find this linkage from his hotspots to the neighboring communities. And I wonder if that's an opportunity, if there's really a epidemiological and social difference, or if maybe there's something to ask about the way some of the techniques are being done, and whether or not that represents your approach to the genetics analysis as opposed to real difference between those types of communities. The second issue is more overarching. I think Christophe mentioned that in the end, you really need to be working in the cloud. You're going to need the cloud for the data analysis associated with this very data-intensive approaches. And yet I find in many African countries and all over the world, and including in the United States, people want to have their data, if they sense that this is sensitive data, they want to have this data on their computer, in their shop, and the idea that, so there's a trust issue with the data governance for data that's this sort of big data that you're generating that's needed for these types of, and I just wonder how different of you are dealing with that, particularly with respect to the governments, as well as the citizens and other community factors that may be worried about data going off into the cloud.
Question in the back.
Hi, Sarah Winterhalter from ICAP at Columbia University. This question is for Dr. Poteet, and it's somewhat related to the funding question, but I wonder, how can we use limited resources and this desire in the public health and biomedical community for these very targeted interventions to address these syndemics, which seem to be so multifactorial and complex? Thank you.
Thanks. And the last question.
Ken Mayer, Boston. A really interesting panel. This question is for Christophe and Frank, but others may want to respond. The work that's presented is really virtuoso, very, very elegant, but also labor and time intensive. Obviously, the technology is improving, but I'm just curious with your crystal ball about how soon do we get to the point of some of the conundra that Gail mentioned, where you actually can have more real-time interventions based on the information generated, because there were several year lag for some of the experiments and studies that you were describing.
Let's go to the panel, but I just have one additional question, but related to the one we heard in front from the first question we heard now. And I feel that one of the key aspects of the issue of trust with putting the big data on the cloud and so forth are two types of certainties or uncertainties. The one is, how certain can we be when linking transmission pairs? That's one issue, Christophe, you commented on, but perhaps you can comment more on that, especially in the context of that issue of trust. And the second is the certainty of the anonymization of that virus genetic data.
Am I starting? Okay.
Can we be really quick?
I'll be quick. We can talk afterwards, but I think it's a matter of prioritization and political will, as I mentioned earlier. And I think we've seen study after study that, at least recently, if we think about the voice trial, for example, where it quote-unquote failed, but what happened is there wasn't a broader understanding of the social conditions and the social factors influencing the study, as well as influencing the outcomes of the study. So I think if we can remember those lessons learned from previous research and how important it is to incorporate those types of conceptual models in how we think about studies, and have the funders prioritize the importance of having that multidisciplinary team, then I think that would be that would go a long way.
Okay. I'll start with the crystal ball question. How long before it's real time? Well, I can say there's a number of grant funding initiatives about transforming these labor and time intensive studies into real time. So I would say it's 2 years before it's sort of technically feasible. I would say we're already at the stage where what we were doing in a labor-intensive work 2 years ago is being reduced to sort of running robots and software. In terms of linking that back to the interventions, again, we're looking at funding opportunities. Work is funded to use that. Maybe less in the case of HIV, because some of the aims in HIV are to extract broad generalizable knowledge, but certainly in other viruses and in outbreaks situations. So I know that multiple groups are funded now with mobile labs and mobile data centers effectively to use that in real time, ourselves included. The issue of the cloud and of trust, I think, is a really hard one. I can tell you our solution. It depends on the setting. One is for the data to stay local at the sites and be interrogated. Essentially, you get the software to move, and so the person at the site has consent. In HIV, actually, that's not considered desirable always because of the issue of criminalization and the idea that the data could be open to sort of police requesting it and so on. So in that case, what we're doing is we're keeping the data off-site in a secure high-performance cluster that people can log into and can't move the data. And essentially, there's the lock, and we have an accreditation system where people sign up to a code of consent. What I'd like to say is actually some of these issues go way beyond phylogenetics. I mean, cloud-based solutions are sort of entering the whole of healthcare. And we're very used to the idea that, for example, a clinician can log in to a secure cloud-based system and access all of their patient records and all of their records of how they visited different hospitals or the care they got. And the reason we trust it is because we trust in the clinicians. The clinicians are accredited, and if they misuse that data, they can be penalized or struck off. And I think some of the same concepts, thinking about this work in a clinical and a public health basis, the same concepts of accreditation and trust, and making sure that we build a community of people who use the system in a trustworthy manner, to me, and adding in sort of data security and so on, it feels like the right way to go.
Do you have any quick comments? Because we've only got 1 or 2 minutes left. Frank or Gail?
Maybe I can just quickly respond to the question about the sort of comparative analyses and the fact that we're seeing different things. I think there's a lot of power in comparative analysis, and it is interesting because one would expect that if there was sort of complete open mixing between these populations, that the hotspots would disappear eventually. And we're not seeing that. So in our area, there's obviously there's a closed network there to some extent. It might not be closed by geography, but it might be closed by risk, for example. So Christoph's group and my group are already talking. We have a paper plan. So all I can say is watch this space, and we're looking forward to that comparative analysis.
Yeah. And I just wanted to say one final thing. The gentleman from the CDC raised the question about governance, and I think that's just absolutely critical. Trustworthy governance is something that's been hotly debated in the United States with regard to where genetic and genomic data are placed from NIH-funded studies. And there's a tremendous amount of distrust within NIH investigators for the cloud and what will be protected and what can be, who will get access to the data. In addition, the H3 Africa project, which some of you may have heard of, which is an NIH-funded project to improve genomic research in several different African settings, has also suffered from the difficulty of on-the-ground researchers having access to and being able to conduct the kind of research that was intended. Therefore, a lot of data leave, to the great consternation of local researchers. So I think when the model that Christoph described, and I think that Frank, who lives and works in Africa, described, can be taken as really sort of good examples of establishing local trust. But there's going to be a lot of other kinds of research that may not take those as their marching orders. So I think trusted governance is something to always watch on. Thank you.
Okay, it falls to me to make some closing remarks on this really fascinating session. A bit like a traffic light, red, green, and amber. So Tonya said go beyond the individual level, go to interventions. Christoph said go to the car park if you want yourself published. And Frank clearly told us not to go on the national road if you want to avoid infection. So we had some intriguing insights. Pictures that I perhaps haven't seen before from this data with this next generation sequencing, and this nice balance that at the moment it's epidemiology and not forensics, and I think that's a real message we should take from your data. We also look at ancestry possibilities. On the plenary yesterday we had all the ancestors. We had Elizabeth Taylor's grandchildren, we had Omar Sharif's gorgeous grandson, and now we've got the real ancestry in the lab. Thank you, Frank. And in a way, I suppose everyone gave us a window. There's a window of opportunity with syndemics. There's the genome window that you described. Frank, there's the knocking on the window to test everyone in your entire centres. There's the corridor of clusters, a nice image. And finally, I think the overarching message from Gail is think about what we all know, and ethical approaches will stand us in very good stead to do wonderful research and will get us into trouble if we don't. Thank you. Thank you, everybody, for a really wonderful session.